Hello, my name is Kathy Yelf. I'm the Chief Executive of the Macular Society and hosting this webinar tonight. Thank you very much indeed for joining us. As ever, we're about to hear a fascinating talk from tonight's guest, and there will be time for your questions at the end. So please do use the chat function throughout the talk if you like to put your questions, and we'll come to as many questions as we possibly can at the end of the talk. Now, what do you think you have in common with a zebrafish? Zebrafish are popular freshwater aquarium fish. If you're a fish lover, you may have an aquarium. They're quite small. They're part of the minnow family, and they originate from South Asia, basically. So what on earth can a small minnow-like fish tell us about macular disease? My guest tonight, welcome, is Dr. Takeshi Yoshimatsu, who is Assistant Professor at the Department of Ophthalmology and Visual Sciences at Washington University at St. Louis in the USA, from where he is speaking to us this evening, afternoon his time, early evening our time. But he's also a visiting research fellow at the University of Sussex here in the UK. Now, Takeshi is one of the researchers being supported by you, by donors to the Macular Society, whose generosity helps fund so much important macular research. Takeshi is working on the function and development of the visual systems of vertebrates, animals with backbones, your school biology will remind you, and for this he mainly uses zebrafish. His current focus is on discovering how the macula forms and why this important part of the retina seems to be vulnerable to neurodegenerative diseases like age-related macular degeneration. And he's looking at whether cells from the zebrafish can be manipulated to create a macula because their eyes actually have a similar structure to the human macula. Who knew that we had so much in common with the zebrafish? So I'm going to hand over to Takeshi straight away to give us his talk about his work on zebrafish. Takeshi, welcome to My Macular and Me. Thank you very much indeed for giving us your time. And I'm going to hand over to you for your talk. Thank you.
Thank you very much for your very kind and actually, exciting introduction. I'm very, excited to give a talk about my kind of recent research, and I think it's quite unique. As Cassie already explained a little bit about, the research using zebrafish. So I hope you can see the slide now. I think that's the presentation mode. So yes, it's about understanding the macular diseases insight from zebrafish, and then you might think that what's zebrafish, to do something to do with the macula. And I'm hoping that by the end of this talk, you might be convinced that maybe actually we can understand what's actually happening in the macular disease using the zebrafish. And then one of the points that I actually want to make in this talk is that using the animal model is very critical for not only just understanding how the disease happened, but also eventually you need the animal model to test the therapeutic strategy, for example, testing a drug, or what you understand what genes actually causes a different type of macular disease. And then the limitation that we are actually facing right now is that 95% of the animals using the research is mouse or rats, like basically nocturnal rodents. And then because those animals don't have macula-like structure, basically we are only limited to use the, at the moment, like primates to do the study about the macular disease. And or some cases, people use some like large mammals, like dogs or cats. But my recent research actually discovered that the zebrafish do have this similarity, the part of the eye that has a similar to human macula, and we can actually use that to study using this very small animal that can manipulate the genetics of this animal to understand, get the better understanding of the macular disease, and then in the future, we could use that to test the drugs. So just enough of this introduction. I'm gonna just give you an overview of the talk. So the first my research is more focused about the neural circuit in the macula, because this is what eventually what matters. Basically, the reason people see from the macula is because there are neural circuits there. Right? And then we try to understand why then this macula is sometimes specifically affected by diseases. And in fact, this is the region of the eye that is most, affected in many diseases. And then I'm going to talk about the result from our recent research and what we kind of, using the zebrafish, what we actually found out, what new things we learned from zebrafish. And then specifically, I'm going to talk about this one molecule called the retinoic acid, and then why this could be a key molecule that affects the sort of like a disease or maybe healthy condition of macula. And then finally, the outlook. Basically, this is still ongoing research, so we still don't know whether retinoic acid is actually good or bad. But hopefully, you will be convinced that the understanding what retinoic acid is doing is important for the future, to understand how to maintain the healthy macula. Okay. So this is sort of like, if you examine the human eye, the overall structure, looking at the overall structure, examine the overall structure, then you will find the macula. But if you examine the macular region more closely and look at the neural circuit, there are lots of differences compared to the rest of the eye, which we call it periphery. So the macula has a very specific neural circuit and then occupied by specific neuron compared to the periphery. So I'm not going to go too much detail into the neural circuit, but there are a few things I want you to sort of remember. First of all, retina is composed of 3 layers of cells. The most outer part has a cells called photoreceptors. So those are the cells that are responsible for detecting light. And the next cell is the next layer is composed of the neuron called the bipolar cells. You don't need to remember the name. And then last layer is composed of the retinal ganglion cell, which then project to the brain so that we can eventually see the vision. So today, we are going to mainly focus on the photoreceptors. So the photoreceptors, there are 2 types. One is called rod, which is responsible for night vision. So the rod is very sensitive photoreceptors, and it only works during the dark condition, such as during the night. Whereas the other type of photoreceptor called cones, those are responsible for daylight vision and then also responsible for color vision. And as you can imagine, macula is very much concentrated by cones and then very sparse rod, whereas the periphery has lots of rods and then little bit of cones. So then the reason the macula is so critical is that those are the places the cones are densely packed, and then that's the vision for daylight. Right. So then if you have the disease for macula, you will lose the vision for the daylight. And then that's why we need to protect it. Okay. And then there are not just the density difference. Actually, the shape of cones are different in macula. So the cones are not only very concentrated in macula, but also it exists in periphery. In macula, though, the cones size are smaller. It's more like actually the photoreceptors are sort of a columnar shape, but then this column is thinner in macula compared to the periphery. Periphery's cones are much thicker. Another difference is that the photoreceptor has this sort of like a compartment in the cell that is responsive for detecting light. And this compartment is longer. So remember, the photoreceptor is a columnar shape. Imagine top half is the bits that detect the light. And then this top half is longer for the cones in macula compared to the periphery. So there are these kind of structural differences exist in human macula. Then the question is, so is that structural difference kind of then sort of affects why these cones in macula are more vulnerable to diseases? What are the physiological differences that macula's cone has compared to peripheral? Those are the questions actually we don't understand. However, we can use zebrafish to look at the detail of the physiology and then how maybe we can explain why these cones in macula are more vulnerable. So here, like a zebrafish, actually, we've recently found that has a region more like a human macula, especially about the specific type of cones called the UV cones. And then human macula is actually located at the center of the eye because that's the part that looking forward for the human. Whereas the zebrafish has eyes on the side. So then actually zebrafish macula is located more towards the back of the eye, which is then looking at the front of fish. Okay, so the location is different. But if you look at the structure of the UV cones in this back of the eye towards the tempora of the eye, the structural specialization is very similar to human macula. So if we examine the length of the outer segment in this specific region in the zebrafish, we call it acute zone. The outer segment is longer in this region, and then density is higher. Okay, so then there is some similarity in the structure. So then we can look at the physiology of those cones to see if there are any difference between the acute zone UV cone and the peripheral UV cones. So to look at the physiology, we use these genetics tools, which is very useful and unique for zebrafish. We cannot do the same thing for, of course, the primate or human, of course, or even the big mammals. Basically, this trick, we specifically express some sensor, in this case, actually, calcium sensor in photoreceptors. So here is a little bit of neuroscience, but not too deep into the details. So the calcium is very important for the neural functions. Okay, so the first of all, calcium is everywhere in the body. And then its actual concentration is quite high, but inside the cell, the calcium concentration is very low. It kept very low. And that is very important for the function of cells or even neurons. And then to keep the calcium concentration low, cells are constantly pumping out the calcium, which means that actually it takes energy to pump out the calcium. Okay, so that's the kind of the basics. You need to remember keeping the calcium concentration lower takes more energy. But this is actually important when neuron is working. So what neuron does is that probably you might have heard of it. Neurons get the action potential. Neurons fire, which means that basically when neurons are activated, for example, by light, then it has a voltage change. What voltage change comes from is that there is sort of a channel that opens when the light hits. Then the calcium goes into the so that this channel actually let the calcium go through. And remember, the calcium concentration is higher outside and lower inside. So then if there is a channel open, what happens, the calcium rush into the cells, right? Then that causes a sort of an electronic current. Then that causes the voltage change. So that's how the neurons are active. Right? And then once the calcium goes inside the neuron, what happens is that triggers the sequence of reaction and then causes the neuron to release the chemicals called the neurotransmitter. And then this chemical is detected by the next neurons. And then this neuron get activated and they send the signal and to the next neuron and then so on. And eventually, when you light it detected, the whole signal cascade goes through and then brain knows, oh, there is a light. Okay. So by looking at the calcium concentration, we can kind of understand like what the state of the neuron. Okay. So then we express this calcium sensor in zebrafish. And the one thing in specific UV cones, and the one thing we immediately notice is that calcium concentration is higher, always higher in this acute zone. What does that mean? Basically, that means that you need to always pump out the calcium because calcium is always coming in. So then you always constantly have to pump up. Whereas the rest of the eye, like a periphery, calcium concentration stay lower normally. So then pump can rest a little bit when there's no light. Okay. So then that kind of suggests that actually the UV cones, the acute zone is not just have the higher concentration of cones, but the cone itself is also very energy demanding. Okay. And also we look at the sort of like a kinetics of cones. So kinetics, what does that mean? So how the cones respond when there's a light, right? So then when there's a light, as I explained, the calcium goes inside the cells and then there is a response. And then eventually calcium is pumped up, okay? But then this pumping out is slower in acute zone UV cone. This is what we found, which means that the pump is working for longer time to actually clean up, which again suggests that they're using more energy. Right? So then it is known, another fact that I want you to tell is that the cones are the most energy demanding cells in the whole retina, whole eye, including the rest of the neurons, including the macular glia, other cells. The cones are the most energy demanding cells. And the macula, because macula has the concentration of cones, right? So which means that the energy demands is higher. So then rods are also photoreceptors, and they also demand the similar kind of like energy demands, but then compared to cones, it's much less. So already, because of the higher concentration of cones, macula is very, very energy demanding. But then what we found from zebrafish is that cones in the macula is even more energy demanding, right? So then that might cause some problem if there is some sort of disease condition that energy supply is short, then those cells start to starve fast and then maybe, die fast. Okay, so that's kind of like makes sense, right? If you have the cells that are very highly demanding, then if there's a disease happen, those are the ones that dies fast. Then the next question is, well, can we actually do something about it? So then what we kind of focus on next is the signal called the retinoic acid signal. So I'm gonna explain briefly why we focus on this signaling. So you might heard of this molecule called the retinol or it's also called the vitamin A. This is very known, right? The vitamin essential for vision. So vitamin A is kind of like processed in the body, and then that will be used for the visual pigment. So that's the pigment that detects the light, right? So the vitamin A derivatives are used for visual pigment. But actually, vitamin A is also converted to some specific molecule called the retinoic acid. And then this retinoic acid is act on the cells to change the gene expression. So then that is more direct effect on many cells. And one of the cells that might be affected is the photoreceptor cells. And then what is more important thing is, so there is this enzyme called the CYP26, which degrades the retinoic acid. Okay, so there is an enzyme to produce the retinoic acid, and then there is an enzyme that degrades the retinoic acid, so that it balances, they both together balance the concentration of retinoic acid. What is interesting, or what is kind of reason that we focus on retinoic acid is that this degradation enzyme called CYP26 is specifically exists in the macula. And that's the same for fish, same for human macula. CYP26 exists in the macula or zebrafish acute zone, which means that the retinoic acid concentration is lower. Okay, so then we suspect that maybe retinoic acid being lower concentration is important for having these specific cones in the macula. So then we did this experiment in zebrafish, right? So then what we did is actually block the retinoic acid synthesis, which means that across the eye, retinoic acid concentration is lower. So then our prediction is that everywhere in the eye becomes like a macula. Okay. And then in fact, what we found is that is the case. So what we found is that the outer segment length in the UV cones became longer in the periphery. And then the acute UV cone outer segment was already long and it stayed the same. Okay, so that means that actually keeping the retinoic acid concentration lower is important to keep the acute zone UV cones outer segment longer. Okay, so what does that mean, right? So then, yes, to have the proper function of retinoic UV cones in the acute zone, you need to keep the retinoic acid length concentration lower. But then, would that actually mean that the keeping the retinoic acid concentration lower, making the UV cones in the acute zone more metabolically demanding, right? Because as I told you, this structural specialization making the cones in the macula or acute zone in the zebrafish more energy demanding. So then next question would be then, would that help if we give more retinoic acid so that the outer segment length kept shorter, so that if the retina becomes in this condition, get into the condition where the energy supply is short, maybe we should give the retinoic acid so that the cones outer segment lengths are shorter, so that they don't stop. But then you can also think that maybe it's actually going to act in the other way around. Maybe if you give the retinoic acid higher, then the cone outer segment becomes shorter, but then the function of the cones are sort of disturbed, and then cones are actually not so happy, right? So this is actually kind of an important question that we are trying to figure out next. Like, we now know that the retinoic acid is acting on the cones to make it more like acute zone-like UV cones. But then if you change the concentration of retinoic acid, would that help when the retina is in sort of a, disturbed condition? or make it worse. And then why this could be useful, and also there's some implication, is that the retinoic acid, as I explained, is a derivative of the vitamin A. So there are cases where you don't have enough vitamin A, or there are cases you have too much vitamin A, right? And then maybe that might affect the health of macula. Also, retinoic acid by itself is used for treatment for other things, actually. For example, some skin disease, retinoic acid treatment is used. So there is a cream, or actually you can even buy a cream that contains the retinoic acid. If you just go to the Boots or somewhere, you can find those like a facial cream, and they advertise it contains the retinoic acid, and it is good for skin, right? And then because the concentration in those creams are retinoic acid concentration very weak, so we don't expect much effect in the eye. But then basically the point is that we are exposed to retinoic acid in many ways. So then by any chance we exposed to the higher concentration of retinoic acid, would that actually do something about macula, right? But we don't know. So that's where we want to understand more, like how may the retinoic acid exposure might affect the health of the macula. Okay, so that's the kind of the summary. But then, I only talk about how the retinoic acid acts on the photoreceptors, but we actually don't know about the rest of the neuron I just briefly touched on, which is the biparcel ganglion cell. And then those neurons are also different in macula. So maybe there is also, the retinoic acid can do something like, act on the biparcel ganglion cell, so that maybe the macula is sort of becoming more susceptible or maybe more strong. That's another future direction. And then finally, as I said, right, using the animal model, I hope you are convinced that it's very useful, because we can do the experiments. You can treat the gene, express some genes, or you can treat with some drugs and then see how it changes. And the zebrafish is just one model that we've recently found that there's a macular specialization. But there are other models that has more like a specialization similar to human macula. And the one of the animal we are kind of now exploring is a lizard. Probably this is also a surprise for you that why lizard is something to do with macula. But actually lizard has a very, very developed macula-like structure. And then, because this animal is easy to keep and abandon, maybe we can use it for experiments easily. So with that, I want to finish my talk by thanking the people who are involved. So the data, like a talk, the topic I talk about is mostly done at the University of Sussex. And then the first part of the talk was funded from the various funding source. And then the part that I talk about, which is the retinoic acid part, is funded by Macular Society. So with that, I'm happy to take questions.
Takeshi, thank you very much indeed. That was an amazing talk. Thank you so much. Now, we've got some questions coming in. The first one is the name of the neurotransmitter at the nerve endings around the macula. I don't know whether that was just not heard, but if you could just repeat that a little bit. What are the what is the neurotransmitter?
Yes, so the neurotransmitter is called the glutamate. So that's all this is same for the human or zebrafish.
Okay, thank you. So your questions in the chat, please. That was quite scientific. And I'm not a scientist. So a question here about zebrafish and how you found out. How was it discovered that zebrafish have similar eye cells to the human macula? How did you discover that?
Oh, thank you for asking. I can talk about all night, but to just make it simple, Actually, we were not trying to find the macula in fish. Actually, we are more just interested in how this, vision works in zebrafish. And then we are kind of surprised to find that actually there's lots of similarity between the zebrafish eye and the human eye, and then that doesn't exist in mouse. And then it's kind of easy to explain though, because the mouse or rats are nocturnal animals, right? So then if you think about the place they live, it's very different. Whereas the zebrafish is, live in the daylight condition, basically the same as humans. So that's probably, maybe it's not so surprising that we found that, the macular specialization. It's more surprising that people have been using mouse or rats.
More surprising that they've been using a mouse. And it is a problem that there are not very many, as you pointed out, other than the primates, the higher primates, and nobody wants to experiment on primates, do they, if they can possibly avoid it, that have actually formed macula. So there aren't, the animal models aren't great, are they, for any of this? And of course, this is one of the reasons I think why a lot of people object to using animals in the first place in macular research, because they don't have maculae, so it doesn't translate. But it's a difficult thing to avoid completely, isn't it?
Yeah, exactly. I think that's one of the biggest limitation about understanding this disease.
So a question here is, what is the role of zinc in the macula, and is its absorption influenced by RA? So we know that zinc, for example, was put into the ARIDS formula supplements, wasn't it? For example,
yeah.
It might be beneficial. Can
yes.
you tell us more about that?
Yeah. So as far as I know, I don't think there's any link known between zinc and then retinoic acid synthesis or degradation. So probably there's some kind of maybe indirect interaction, but in terms of the context of a retinoic acid, probably it's an independent thing. So then it's, I think it's important to understand like the both basically.
So it's really interesting that increasing or reducing the amount of could go either way. We don't know yet whether what's going to be beneficial and what actually might be harmful, and what might be beneficial for one layer of the macula might be disbeneficial and not beneficial for a lower down. So how do we approach that? How do we go forward from that?
Yes, so that's very powerful about zebrafish. So we can actually, first of all, right, we can treat the fish with retinoic acid or deploy the zinc, and then we can look at the UV cones or next neurons by, for example, using the same technique, right, using the calcium sensor to see the state of the neuron, how much they use the energy, or actually there is more direct way to measure the sort of energy demand in cells. And those are the technique actually you need to manipulate the genes to do that. And then zebrafish is, I think, one of the only actually animals that we can do this kind of experiment, and then also has the macular-like specialization.
Yeah. So another question about macular dystrophies. So we've been talking specifically about AMD, age-related macular degeneration tonight, but we know that retinol, vitamin A, these are issues in more than one condition, aren't they? More in other retinal diseases. So what is there a possibility that zebrafish might be useful, or are they indeed perhaps already being used in some of these other dystrophy conditions?
Yeah, absolutely. I believe this is going to be the next big direction. So again, like this is a very recent finding. And then there are many studies actually to understand the genetic sort of contribution of macular disease. And then people sort of like, look at the genome sequence of the people who has the macular disease. And then we kind of have the good idea about the candidate genes, right? So then there's a risk factor of the genes, problem is, how do we test it, right? We cannot then make this gene not, removed from the primate. Right? But now we have this system that we can easily make this removing particular gene to see how this macular specialization is affected. And then this is the interesting thing. So the many candidate genes that we got from the human study, many of them are involved in the metabolism, which suggests that actually energy supply is the key part of causing the disease.
And when we talk about energy supply in eye cells, what is the energy? Are we talking about oxygen? We're talking about key micronutrients and things like that. What are we talking about?
Yes, actually combination of all. So the oxygen is a very big part. Another part is the ATP. So that's basically the energy, it's the fuel for the cells. And the ATP is basically sort of like supplied by, by the chain of the reaction. So it's just many things. And then now we actually have the tools to look at the different sort of like metabolic molecules, ATP, oxygen, or other molecule, for example, glucose or lactose, and then we can look at those consumption in the live animal in zebrafish.
And do we understand why? Maybe you explained this and I missed it. Sorry. Do we understand why the cells of the retina and the macula in particular are so hungry for this energy? Why?
Yeah. Right.
They describe it as the most metabolically active or demanding organ or something like that, don't they? So
yeah.
do you know why would that be?
Actually, we know about it. So that was a kind of like, my previous main focus of my previous research, and it's very quite, detailed. But basically, what macula needs to do is not just having this highly packed cones so that you have the better resolution, but have to be active all the time, up and down, like a neuron can be active or non-active very fast, so that we can see the sort of time resolution better. Right? So then to do that, neuron basically kept the calcium level higher so that it always go up or down either way very quickly. So we know, right? This is like energy demanding, makes cells susceptible, but because of the macula is so important for vision, they have to do it.
Okay, so I once heard of somebody who had been told that their macular degeneration was a result of wear and tear on their macula.
Yeah.
And this person interpreted this as being that they were kind of wearing their eyes out. They were using them too much. If you and spent a long time of the day, this is a misunderstanding, but spent a long parts of the day with her eyes closed and not reading or watching television because she thought she might be wearing her eyes out. Perfectly understandable when you're given this, wear and tear. You do think of that, don't you? But I don't think. Am I right in thinking that actually at night this metabolic rate doesn't actually slow down? It doesn't. So not using your eyes, closing your eyes, sitting in the dark doesn't make any difference to this metabolic rate or this energy demand.
Yes. So this is actually, we don't really understand. So there's an experiment where people look at the energy consumption in the dark or daylight. And it is true that in the daylight, energy consumption goes down, actually lower. And then the consumption higher in the dark. But when you are sleeping, for example, you close the eyes for a long time. Then what happens is that the photoreceptors slowly adapt to the dark condition so that they don't use energy so much. So if you are in the dark place in the short time period, energy consumption actually goes up. But if you are in the dark place for a long time, we actually don't know how photoreceptors behave.
but it won't be helpful to just sit with your eyes closed in the dark for a long time. Wouldn't necessarily help. Okay, here's another question. Since there are more cones in the macula, would one expect degeneration to affect? Would one not expect, I beg your pardon, degeneration to affect color perception? So are rods more likely? Do rods detect color more?
Actually, the other way around, because the cones are responsible for color perception. Maybe I explained I didn't explain well. So cones are responsible for color perception, which means that the macula is damaged, you will lose the color vision. Like your color vision is going to be impaired.
Okay, right. That's very helpful. Two questions here now. Will excessive alcohol intake interfere with RA metabolism? And the second question of this person is, what percentage of energy is used by the macular mitochondria? So the mitochondria are, would we describe them as the battery pack in the cell, do we? Is that how we?
Yeah. So, okay. So the first question, alcohol intake. As far as I know, I'm not aware of the sort of like, the link between retinoic acid and alcohol. But, as you know, alcohol would affect many things, right? Maybe affect like energy supply. So, it's hard, like lack of direct link does not mean that, those are sort of like, nothing to do with each other. The second question, yes, the energy. So the mitochondria is the one that create the energy. Right. So then that's the same for all cells. I think what is more actually critical to understand is where the cells use the energy, right? So then if you stop that, then mitochondria does not have to work so hard. So that's the, because then if you, for example, imagine, maybe the other way around to think about is to give more energy so that maybe make mitochondria stronger. I think that could be a one strategy. Then the way that we are thinking is to like make the macula use like less energy when the supply is short. That would be our thinking. But yes, it's true. Like maybe you can make the mitochondria stronger so that maybe they are more active. Yeah.
So we know that some research is going on, isn't it, to look at exposing the retina to certain light wavelengths, and in certain areas it's thought that the certain exposure to certain types of light may in fact recharge the mitochondria in some way or make them a bit stronger. So there is work going on to try to recharge those mitochondria.
That is very interesting. In fact, I wonder like how this light changes like the property of mitochondria.
I think we've got to have a webinar on that, I think, in a future event. Another question then is about nicotine. So if we don't know about alcohol, what about nicotine? We do know that nicotine is very bad for your eyes and is very strongly connected and linked, isn't it, to macular degeneration?
Yeah. I think nicotine is so basically more about like energy supply, right? So it's so the retinoic acid is more like not about the like energy, right? Retinoic acid is more about changing the status of the cones. So I think that's basically two things you have to think about, right? Always having the good supply of energy. Right, and then that would be the related alcohol, will be related to nicotine, or maybe something, right? The other thing is that them keeping the cone function normal. So that's the like vitamin A, retinoic acid. So it's a two different things. Of course, it's connected, but it's a two different things.
So the other sort of so-called modifiable risk factors for AMD are thought to be, for example, a lot of exposure to blue light. People are often interested in whether there may be some light wavelengths that are good for our eyes and some are not good, and blue light is often talked about as not being very good. And I guess the other thing is general diet and the absorption of antioxidants and And there's carotenoids, particularly like lutein and zeaxanthin, are often talked about. So what is the potential way in which that they could be affecting? I mean, certainly eating a lot of some I think conditions where an excess, as you mentioned, of vitamin A is considered quite bad, that people are encouraged not to eat a lot of liver, for example, aren't they? Because of that. But where does that all interconnect, do you think?
Yeah. So first of all, keeping the good energy supply, keeping your body healthy is certainly a good thing. And then just be mindful about avoid this sort of excess amount of vitamin A or retinoic acid, because we just don't know how it affects. And then it's just moderate is always like the body can handle it. And then there's many things, same for many things. There's a good side and bad side. You cannot just go one extreme and then expect to be magic. I think that would be the by studying the retinoic acid, I think that was the sort of take home message. If you really remove retinoic acid, that's very bad for body. But then keeping the concentration lower was essential for keeping the macular cones functional. So like just right amount is the kind of key.
Is the balance. What my mother used to say, everything in moderation, she used to say. Here's another question around oxidative stress factors affecting the macula. And this is a phrase I don't understand. Ask the questioner to forgive me, but glycation end products. I think you might explain that to me. Sorry.
I see. So yeah, advanced glycation end product. I'm not familiar about this word, like glycation end products.
Perhaps we'll ask the questioner to help us with that. But oxidative stress is certainly thought to be part of the problem, isn't it, with macular degeneration? So the effect of free radicals, and this is where the diet comes in. If you're not eating a lot of antioxidants, then it speeds up the aging process, doesn't it? Oxidative stress, effectively.
Definitely, yes. And then, of course, like oxygen is, the essential for the mitochondria to make the energy.
So where do you go next then? So zebrafish, are you in zebrafish for the long term? Do you think? Do you think there's got a lot more to tell us, zebrafish?
Yes. So basically, what I'm thinking is the two ways. So zebrafish is very powerful because we can sort of manipulate the genes so that we can do the experiments such as removing the particular gene, right? The known risk factor, and then see what happens. So that's very useful. But zebrafish, as I emphasized, like it has this sort of acute zone that is similar to human macula, but it's not the human macula. Okay, I have to be clear about it. There is a similarity, but it's not the human macula. And then there are other animals that has a sort of like a much more similar place, like macula-like part of the retina, and then that basically we have to sort of expand our options. So as I said, like 95% of people use mouse and rats, but we have to make use of the different animals that we available.
There'll be people who don't agree with using animals at all, of course. How useful are the alternative models, do you think? So using induced stem cells, for example.
Right. So I think that there is a big potential about the induced stem cell, and then people are trying to make the sort of like a retinal tissue from the stem cell. Right? So the current limitation is this: because we don't know how the macula is generated, we don't know how, because so the reason we don't know how the macula is generated is because we don't have the animal model. So it just goes back. So I believe that there's this, stem cell is going to be useful, but we have to step back and then to make the stem cell research more useful, we need to understand how it's actually generated in the animals.
Of course, humans are not very keen on giving up their maculae, are they, when they're alive, to help with this problem? Actually, a relevant question has just come in from somebody who says, or two questions actually. Do zebrafish see in colour? That's one really interesting question. I don't know. And the second one is that zebrafish are able to heal their macula, and that's why they're being studied. Is that true?
I love these two questions. First of all, does zebrafish see colors like human? Actually, they do see colors better than humans.
Wow.
Yes. So yeah, I mean, probably, I could talk about it like hours, but yes, that's the short answer. The second answer.
How do you know that? How do you know that?
Right. So actually, so zebrafish, so the human has probably we know like human sees the three colors, red, green, blue. Zebrafish has three colors plus one more called the UV. So they have the four colors. And then that already suggests they have a better color vision. And then in fact, it's very funny. Like, for example, if we hit the UV light, with my eye, I don't see anything, but fish actually freaks out. And so that's, yeah. Anyway, and the second thing, yes, why can we heal the macula? So that's actually the big potential I'm thinking for the next, in the future. It is true that the zebrafish can heal their eyes, but we don't know whether they can heal the macula, right? So let's say if you damage the macula, can fish actually make the macula, or fish just make a periphery? Right? That's actually, we don't know it, but we do know that they can, heal the retina.
That's amazing. So, in terms of your work in the US and how it interfaces with what's happening in the UK, is this one project, or are you doing different parts of the same project at different universities? How does that work? Or have you now relocated to St. Louis, sort of signing up?
Right. So basically, this project supported by Macula Science, it's a collaboration between myself and then the one group in the UCL. And then I think nowadays it's kind of common. So basically, we use many different techniques. So then different group has different skill sets, for example. So like, for example, myself, my skill set is looking at the physiology of the zebrafish. And then the collaborator has a skill set of looking at the structure or manipulating a gene, right? So as a sort of like, we have to work together to have this complete understanding. So that's how I see it. So
international collaboration is critical, isn't it, now in the research space? Nobody can do it by themselves effectively, can they? It's
definitely. It's just that nowadays, we have to use many different skills, and then it is almost impossible to have everything in one, group.
And how does working in the US compared to working in the UK?
Ooh, that's a tricky question that I did not expect. In terms of the research, I think it's very similar. Of course, like a lifestyle-wise, there's a different, big differences. But in terms of like, in working in the laboratory and then doing the science, it's very similar.
Well, Takeshi, thank you so much for joining us tonight.
Yeah, my pleasure.
Really fascinating and interesting talk, and I'm so grateful to you for giving up your time all the way from the US. We wish you the very best of luck with your researches, and I hope you'll come back at another date and tell us how you're getting on with it, because we're all agog really at the extraordinary work that you're doing. So thank you very, very much indeed for joining us tonight. Thank you everybody for joining us tonight. Thank you for all your lovely questions. Don't forget, please, that this recording, this session is recorded and will be on our website in a little bit, along with all the other My Macular and Me sessions, which are on our website for you to look at again, or catch up with anything that you might have missed, or indeed, please Do recommend them if you found them useful and interesting. Do let other people know if you think that they may be interested or may benefit from learning more about their own macular condition. That would be great. So you go to our website at www.macularsociety.org. If you have any questions that have arisen about your own condition, don't forget that our advice and information service is open every weekday 9 till 5, and that number is 0300 30 30 111. That's 0300 30 30 111. The number is also on our website. So that's it for tonight, but next month we'll be back, and next month we're going to be talking about biosimilars. These are lookalike drugs that are nearly exact replicas of existing ones, but they are cheaper and often more cost effective for the NHS in many cases. There are biosimilars in many, many disease areas now. And it's happening now because as the first licensed drugs for macular degeneration near the end of their patent time, they come off patent, other manufacturers are allowed to create cheaper versions. Now, this is good all round, is it? Or are you concerned that the new ones might not be quite as good as the originals? Well, all your questions will be answered next month. That is the third Tuesday of the month, and we will be in April after Easter. Amazing. And again, don't forget that you can see them all again on our website. Takeshi, thank you very, very much indeed for joining us again tonight. Thank you for all your questions. Thank you for your company. And I look forward to seeing you again next month. Good night, everybody. Have a good evening.